right, so I'm not sure how much folks have heard about the moss study in the news. We're going to talk about that, but first I'm going to enchant you with some background on moss and why we would be using it as a bioindicator of air quality. Since starting to work with mosses, I've come to realize that they are just vastly underappreciated and even maligned by the typical homeowner. In fact, there's a whole industry devoted to moss killing, if you can believe it. But it is a truly magical time right now here in Portland where people excitedly chatter about moss on the radio and the TV. And we even have a pro-moss bumper sticker showing up on cars in the city, and I'm not even joking. So a moss is actually a non-vascular plant. They're part of the oldest plant lineage, and thus they're distant ancestors of the plants you grow in your gardens. Ancient moss deserves our admiration for helping plants make that transition from aquatic environments to terrestrial environments. So being non-vascular means that the moss can't store water. So like a sponge, it absorbs water when it's wet outside and loses water when it's dry outside, during which time the moss becomes more or less dormant until it becomes wet again. Oops. So if you look at the leaf of a vascular plant, it's fairly complex. What's key to notice is that the leaf tissue is sandwiched between a protective cuticle or epidermis, which means that leaves don't absorb water directly. And as you know, all plants take up water and nutrients from the soil via their roots. So a moss leaf, on the other hand, it's one cell thick. It lacks that protective epidermis. So water is absorbed over the entirety of the moss. A moss also lacks roots and access to soil nutrients. Instead, they rely entirely on the atmosphere for nutrition. So to make this life strategy work, the moss cells have a high density of binding sites for trapping nutrients from rain and dew, which inadvertently also causes the moss to accumulate high levels of metals and other pollutants. So this is why sphagnum, or peat moss, for instance, is commonly used in remediation of oil and chemical spills. You know, moss are just very absorbent organisms, and they're very effective at sequestering metals. So pollutants in the form of particulates, as many of the metals typically are, can also become trapped on the surface of the moss. So what you're looking at are scanning electron microscope photos of particulates on little moss leaves that were collected from different environments. So our measurements of metals in moss include both the surface particulates as well as those that actually get sequestered inside the moss cells. So moss has actually been used to indicate trace metals in the environment since the late 60s, and literally over hundreds of studies. Most commonly, these studies focus on natural areas or mapping in the vicinity around a known industrial emitter. So the study that we're going to discuss is somewhat unusual in that we attempted to map pollutants in fine detail across an entire urban area. Only a handful of past studies actually compare metals and moss directly against measurements of atmospheric concentrations taken by air quality monitoring instruments. But overall, these studies tend to show pretty strong relationships for some of the more toxic metals like cadmium and lead. But on the other hand, some of the other metals, we know a lot less about correlations to atmospheric levels. Relatedly, it's unclear what environmental factors besides air quality might affect metal concentrations in moss tissues. For instance, as Rob mentioned, to what degree do intense precipitation events wash off those surface particulates versus, facilitate their absorption by the organism, which we know rain does help facilitate that absorption of pollutants. We're also not sure what time frame is represented by moss levels, metals levels in the moss samples. So, the literature is complex and often conflicts on some of these topics. So the bottom line is that there is much that we still don't know. So given these uncertainties, you may ask yourself, why open a can of worms and use bioindicators? I mean, we already know how to measure air quality, not to mention all regulations and health benchmarks are written in terms of concentrations measured by instruments using very specific EPA protocols. And so it basically boils down to cost. You know, Portland has one permanent air quality monitor, and it costs about $150,000 per year to run. And about $40,000 of that is spent to measure metals. And for one moss sample, it costs us about $150 in total, including labor, you know, if you pay your employees generously. So this is our study area. The star on this map is Portland's one permanent monitor. For special projects, there may be a few additional mobile monitors in play. But as you can imagine, this is a very cumbersome and expensive way to try to identify sources of pollution, and you can't make maps with such a small number of data points. So in December 2013, we collected 346 moss samples across the city and measured 22 elements, including heavy metals. So with this large number of samples, we were able to look at how pollution is distributed across the city with detail that's never been seen before. So we sampled on a 1-by-1 kilometer grid, randomly selecting a location in each grid cell to collect moss. And we did this rather quickly, all within about 2.5 weeks, and also within 6 traverses across the city. And we did it this way because, you know, there are some studies suggesting that weather could affect metal levels in moss. So if weather is important, then us sampling systematically from one side of Portland to the other would make it harder to separate out these effects in our model. So here's the moss. This is Orthotrichum lyellii. It's extremely abundant in Portland, and in a lot of West Coast cities, actually. It occurs on about 75% of hardwood trees in Portland. So we prep the moss by trimming away the base. We remove the material, insects that make homes in there, whatever debris you might collect in the moss. And then to extract the metals, we use graphite block digestion with nitric acid and hydrogen peroxide. And then we use an analysis called ICP-OES to determine element concentrations. And this is actually a pretty standard lab technique for extracting metals from moss and lichen material. So we looked at cadmium first because the Oregon Department of Environmental Quality has been trying to track down cadmium sources in Portland since 2009. So this map shows cadmium levels predicted by the DEQ, who based this on emissions data from facilities with permits to emit cadmium. So the cadmium map suggests relatively low levels everywhere except for a hot spot you see near our northwest industrial area. However, Portland's air quality monitoring site, so that's the number one by the little red plus on the map, and was measuring much higher cadmium levels than was predicted by the model, so they knew that they were missing something. So just briefly, cadmium is one of the most toxic of the heavy metals. It's a human carcinogen. It's associated with many negative health consequences, including kidney disease, learning difficulties in children. So we built spatial regression models of cadmium and moss, and then we used the model to estimate cadmium on a fine 50-meter grid across the city. So as for important predictors of cadmium in moss, the type of tree we collected from, the weather, the density of roads, tree canopy cover, none of these were important, which in a way is good because it keeps our model simple. To our great surprise, though, distance to permitted cadmium emitters was also unrelated to cadmium levels in moss. However, a very strong predictor was distances to 2 unpermitted stained glass manufacturers, and it turns out that cadmium is a pigment used in making red and yellow glasses. So other significant predictors included distance to the Washington border and land use in the vicinity of the moss sampling site. So here's our map of model predictions of cadmium as detected in moss. Looks pretty different. So we see two large hotspots by the two stained glass facilities, so those are indicated by the numbers 2 and 3 on the map. And then the purple circles you're seeing, those indicate levels of cadmium emissions by permitted emitters. And as I said, there was no relationship, and you can kind of see from the map that there's no relationship between permitted emitters and cadmium levels in the moss. So at this point, we wanted to check ourselves and see if we could find multiple lines of evidence to support our growing hypothesis that stained glass was behind the cadmium emissions. So we pulled out our data on arsenic and selenium in the moss. And these are both used in stained glass manufacture. And we found hotspots of both elements by the southern facility, so that's the black triangle on the map. And that was the larger among the two cadmium emitters. And it actually came out in the news later that the northern facility had stopped using arsenic years ago, which may explain why we didn't also find an arsenic hotspot there. So we became concerned and informed DEQ of our findings, and they placed a monitor near the southern facility in October 2015. And at the same time, we went out with students from an urban ecology class at Portland State, and we collected moss at another 25 sites sort of concentrated in that area in southeast Portland. So our new cadmium and arsenic and selenium data all supported our identification of the glass facility as the source. But remember, all of our maps are relative concentrations of metals in moss, and while we expect that high levels in moss will mean high levels in the air, we really had no idea how high until DEQ got their monitoring data back, which is when everything really started to hit the fan. So what you're seeing here are cadmium concentrations in the air over time, and that's the blue peaking line. And then the pink and green and purple horizontal lines you see lower on the y-axis, those indicate different health benchmarks for chronic and acute exposure to cadmium. So in a nutshell, the DEQ found cadmium levels roughly 50 times higher than the state health benchmark and 150 times higher for arsenic. So at that time in October, there actually happened to be 3 other monitors that were out at the same time, so we were able to do a very rough comparison with moss cadmium concentrations, and we had a very high, remarkable correlation of 0.99. So, while 4 sites really isn't a sufficient sample size, this result nonetheless suggested that moss may actually be a quite accurate indicator of cadmium. So, needless to say, the October monitoring results were quite alarming. The facility is shown in red and the monitor as a star. This area is partly residential. There are 2 schools and a child care center all within close proximity to the facility. Those are shown by the yellow triangles. And it turns out that neither glass facility was using particle filters to trap cadmium and other metal particulates emitted by the glass furnace. So these findings then kicked off a tornado of media attention and caused the DEQ and EPA to re-examine pollution control regulations for the stained glass manufacturer. So to hear what happens next, stay tuned for Katie and Julie's presentation. So the big picture, in my mind, the problem solving and cooperation between agencies and our use of complementary approaches to air quality monitoring was truly revolutionary. This work could be duplicated in any city where moss or lichen grows in sufficient abundance. It bears repeating, though, that the moss data, at least at this stage in the research, must be validated by air quality monitors in order for us to know whether hotspots are problematic or pose, some sort of health risk. So we really see the moss as a useful screening tool to help regulators prioritize where to focus limited and expensive resources like monitors. So Portland is particularly well endowed when it comes to moss and lichen. However, the metals analysis only requires about 0.5 gram of biomass. That's not very much. So that means that many cities across the U.S. will have sufficient material, even if you don't necessarily notice a lot of moss and lichen on your trees. The abundance of moss and lichens in any particular location or city can be hard to predict. So prospective studies will require some on-the-ground scouting. Conditions that are particularly unfavorable include dry, desert-like climate, and areas with high sulfur deposition, which is directly toxic to moss and lichen. But for areas without enough bioindicator material, there are some alternatives that can be considered. The most developed technique is using nylon bags of sphagnum, which you then attach to trees or stands, and then you retrieve it at a later date and analyze the metal content. And what's amazing about sphagnum is it will continue accumulating metals even after it dies, because perhaps the air quality was too poor in the study location. Some folks have had decent luck working with bromeliads. You can also, sort of like the moss bag, you can also transplant other moss and lichen species. And then I've seen a few studies that have also used metals analysis of tree bark. So other pollutants can be measured as well in the moss. Nitrogen, sulfur, mercury, several persistent organic pollutants such as PAHs and PCBs and so on. You can also look at pesticides and herbicides, although that requires a pretty good chemist to be working on your team. And additionally, the use of isotope ratios is a pretty interesting developing field in which isotopes are being used as tracers to help differentiate between different emission sources, for instance, between natural versus mobile versus different types of industry, and what sources might be contributing to levels detected in your bioindicator. So next step for us, as Rob said earlier, we're going to be publishing data this summer along with some basic maps for the remaining 20 or so metals and elements that we looked at. So there'll be more hotspots to talk about soon. We're also looking for resources to do a calibration study to help us sort out some of those fundamental questions I mentioned about, how accurate is the moss and what time frame are we talking about. There's also an interesting opportunity to work with the Cincinnati Children's Hospital where we would compare metals and lichen with neuroimaging and other health data from a large cohort study of children in the area. And then, finally, as we consider our research, our next research steps, I'd be interested also in hearing, the opinions of folks in the audience of what you think our next steps should be, or any particular issues you think that would particularly benefit from the use of bioindicators. And that would be it.